All right, well, good morning, Exphys class. Welcome to the notes for the nervous system. This will be for class this Friday. So we're going to go ahead and dive right into it, and let's get going. So just some very basics about the nervous system. The basic anatomy and setup of the nervous system, kind of already mentioned this. The central nervous system includes the brain and the spinal cord. Brain and the spinal cord are the location of the cell bodies of neurons, right? Or the somas. Okay, so that's where those are found. The peripheral nervous system is primarily axons. Right? Definitely those peripheral nerves are primarily axons. Okay, so it's just a little bit about the setup. Okay, and we're going to spend a little bit of time talking about membrane potential. And so we'll start talking about resting membrane potential, kind of what governs and dictates that. And then we'll move from there into the phases of an action potential. And we'll go from there. Okay, so membrane potential. It's this idea, right, that we have a charge difference from one side of the cell membrane to the other. Okay, and that typically is going to mean, that does mean that one side is more negative than the other, and one side is more positive than the other. Okay, and so when we talk about resting membrane potential, so we're talking about the resting phase, we have a setup that looks something like this. Okay, so, and this is looking at the outside of the cell, right? And this is looking inside the cell. Okay, and this is our cell membrane. All right, so we've got outside, we have just a little bit of potassium, but inside we've got a lot of potassium. Okay, outside we've got a lot of sodium, inside we've got a little bit of sodium. Okay, calcium, we don't really care about too much. Chloride, we're not going to talk about too much. Okay, but the big deal there is that the primary ion of the intracellular space is potassium. Primary ion of the extracellular space is sodium. Okay, so what that means is there's a strong gradient for potassium to go out, and then there's a strong gradient for sodium to go in. Okay, and that's our chemical gradient, right? Just a typical diffusion gradient. All right, and overall, the membrane at rest is more negative on the inside and more positive on the outside. Okay, so why is that? Well, there's a couple reasons. So which side's positive? Outside. Which side's negative? Inside. Okay, there's really two main reasons for why this is. Number one is there's more leakage of potassium than sodium, greater leakage of potassium than sodium. Okay, so if one of those is going to leak, potassium would leak out, right? Because that's the way it wants to go with the diffusion gradient, and sodium would leak in, right? But if there's more leakage of potassium, then there is potassium going out, then there is sodium coming in, then that means we're losing, right? This causes loss of positive charge, right? So we're losing more of a positive charge inside the cell than we would be gaining a positive charge, because if sodium leaks in, that would be gaining a positive charge, okay? So that results in a net negative charge on the inside, right? Because we're losing positive charge, okay? So that's reason number one, why we have more negative charge inside. Reason number two is the sodium potassium pump. Okay, and that's right here. It's also known as sodium potassium ATPase. Okay, it uses an ATP to pump 3 sodiums out and 2 potassiums in. Okay, so this is working against that leakage a little bit, right? It's working against that leakage because sodium's trying to leak in, but this pump actively pumps it back out, right? Yes, exactly. Sodium's trying to leak in, but this pump actively pumps it back out. Potassium's trying to leak out, but this pump actively pushes it back in, okay? But it does this in an unequal fashion, right? There's 3 for every pump, we get 3 sodiums out and only 2 potassiums in. So that is also a net -1 charge for the inside of the cell because we're losing more positive than we are gaining positive, right? That combined with the fact that we're already losing more positive than we are gaining positive with the normal leakage, because there's more leakage of potassium, that is those are big parts of what causes this negative resting membrane potential. Okay, so that's the idea of resting membrane potential. So we're going to spend a little bit of time looking at these two gates, and this is review. Okay, this is review from anatomy and physiology. If you had me, you probably even remember these same images from the textbook, and I took them from the A&P textbook. Okay, so we have these two gates here. Sorry, two channels. This is the sodium channel. Okay, it has two gates. It has an inactivation gate and an activation gate. The potassium channel only has one gate. It only has the activation gate, these big ones here. Okay, inactivation, activation. Okay, so when we look at the resting state, what do we see? We see that sodium gates are closed, but they're closed in such a way that activation is closed and inactivation is open, right? This is open. Okay, potassium gates are closed. There's only one option there. They're either open or closed. Okay, so is there any movement of ions? No, except leakage, which is minimal compared to what we're going to see in a second. Okay, so what maintains resting membrane potential? We just said over there, right? So it's the unequal leakage of sodium and potassium. So it's more potassium leakage. That's number one. Number two would be that sodium potassium pump. Oops, sorry. Sodium potassium pump, which results in a -1 charge inside the cell because it pumps 3 sodiums out and 2 potassiums in. Okay, all right, so that sets us up in resting membrane potential. You can see here we're following along with the voltage graph as well. So that little purple part is where we're at. Okay, so we move from the resting membrane potential into the situation where the stimulus is received. And now is a good time to introduce this idea of a multiple neuron chain, right? So when we go to contract a muscle, what we do, if we're doing this consciously, is we're thinking about it, right? We have a thought like, oh, I need to reach forward and touch my keyboard, right? So that thought can be quick, but it comes more from the frontal lobe, right? So further up in the frontal lobe, probably places where we make executive functioning decisions, like the prefrontal cortex, okay? So there's some neurons up there. So there's a neuron up there. Let's say this is in the prefrontal cortex, right? That's going to send a signal down back to the primary motor cortex. Okay, that primary motor cortex is going to receive that signal on a dendrite. So the axon of the neuron in the prefrontal cortex is going to send a signal down its axon to the dendrites and cell body of the neuron in the primary motor cortex. Okay, and then if we're really being accurate, we're gonna put this axon down. It's going to spread out again. We're going to put another neuron here. Sorry, this one doesn't look as cool as the last one. Okay, this would be another one. And then we'll put this down here to a big muscle. Okay, so primary motor cortex down to a alpha motor neuron. Okay, so again, signal down this axon, activates the dendrites here. There might even be some branches that go right to the cell body, activates there, and we start an action potential here. That travels down, and then that's going to feed into the muscle. Okay, so that's kind of the chain of events of these multiple neuron chains. Okay, so what part of the neuron receives the stimulus for action potential? Well, it's going to be the dendrites and the soma. Okay, that's just the actual cell body. And then where specifically does that action potential begin on a neuron? Okay, well, it begins in a region called the axon hillock. The axon hillock is really in this very first part of the axon right there. We would say it's right there. We'd say it's right there. So there's lots of these sodium and potassium channels there, not the sodium potassium pumps, but the sodium potassium channels. There's lots of those there. So it's a good place to start an action potential. Okay, so the stimulus is received, right? We think about it, goes to our primary motor cortex. Primary motor cortex sends another signal down to the correct neurons to go right to the alpha motor neurons down to the muscle. Okay, and that's going to help us contract that muscle. Okay, so what's the first step in all of those action potentials, right? This happens every time we do an action potential, not just in one spot. So every time we do an action potential, we have this, and this is happening over and over and over again down each of those axons. Okay, so let's talk about this. So the first step is called depolarization, right? And that's shown right here. That's the part where we're moving in the positive direction. Okay, less negative, more positive. Okay, what do the sodium gates look like now? They're open. Okay, both gates are open, right? Both the inactivation gate and the activation gates are open. Potassium gates, closed still, right? They still look closed. Okay, movement of ions, we've got sodium going in, potassium, none. Okay, so crucial there to see that. Okay, next step, repolarization. We can see now it's going back down in the negative direction. Okay, sodium gates or sodium channel is closed. Let's talk about each of those gates. Activation gate is still open. Okay, but the inactivation gate is now closed. So we call this state inactivated. Okay, but now the potassium gates are open. Okay, and so the movement of ions, we have potassium going out of the cell, right? We can see that potassium going out, right? So we'll take it one step back on the depolarization. We're gaining a positive charge on the inside of the cell. That's what makes the membrane go in the positive direction, okay? But on the repolarization side, we're losing a positive charge, right? So that's what makes the membrane go back in the negative direction. And this works perfectly with the chemical gradient that we already talked about, right? There's always more sodium on the outside, so that's going to help it push in. And there's always more potassium on the inside, that's going to help it push out, okay? Sodium goes in faster. That's just shown by the steepness of the depolarization versus a little bit more gradual slope of the repolarization, because it not only has a strong chemical gradient, but it also has an electrical gradient, right? So this is driven by chemical gradient and by electrical gradient, right? Because it's negative in here and these are positive. So positives are going to go towards negative, okay? On the potassium side, this is positive, this is also positive, okay? So that doesn't help, the electrical gradient doesn't help, but the chemical gradient is strong enough to push it out and overcome that electrical gradient. Okay, so that's repolarization. Last step is hyperpolarization. Okay, we see that here. All right, what do we see? So in the sodium channel, we see that it's closed. Okay, we see that we really have a resetting of these gates. Okay, so the activation gate closes and inactivation opens. Okay, and that's crucial, right? That resetting is crucial to allow for another action potential. Okay, state of the potassium gates, early, they're open, but then late, they close. Okay, so what's actually causing the movement of ions here? Or sorry, what's actually causing the change in membrane potential is the movement of ions. Okay, and so the early we have potassium going out, but then late we have none. Okay, so when it gets enough negative, gets down, see it dips down below resting membrane potential. When it gets down there, that causes the resetting of both those gates. Sodium channel gets back to its resting state, potassium channel closes, and then this slowly starts rising up again, back to resting membrane potential. Okay, so those are the 4 main steps. So it'd be great if you could take a second and pause your video and see if you can walk through these 3 questions just as review. Okay, so the ion flow. So which ions are flowing in which direction with each of these? Okay, name the stage of each. Okay, and then the correct order of events. Okay, yeah. So there. Pause it for a sec. Think about that, and then you can unpause it, and I'll walk you through it. Okay. So which ions are flowing in which direction? Obviously, we can see potassium going out here. This one is none. This one is sodium in. This one is potassium out. Okay, so just a good frame of reference. We got potassium out happens twice. Sodium in happens only once. And no movement is occurring once here in this sequence of events. Okay, so name each stage. All right. Well, let's put them in the correct order first. Okay, so the correct order, we've got, so the correct order, we're going to start here with resting membrane potential, right? Because this is a resting membrane potential state. There's no movement. Okay, from there we're going to move to depolarization. So we're going to go 1, 2. All right, depolarization is when sodium rushes in. Okay, now we have these two almost look the same, right? Because we have potassium going out in both cases, but the difference is the state of the sodium channel, right? This is inactivated. This is being reset, going back to being ready for another action potential. So this one comes first, right? And then this one is last. Okay, so we've got resting membrane potential. We've got depolarization. We've got repolarization. Okay, and then we've got hyperpolarization. Okay, so definitely need to memorize that, be able to reproduce that whole sequence of events. Okay. All right, so just a couple things about action potentials. Action potentials, they are governed by something called the all or none principle, which means we don't have any variation, so no variation in amplitude of the action potential wave. Oops, sorry. Siri, I don't need your help. Thanks, though. Okay, sorry, guys. One sec. Okay, and we're back. Sorry about that. Siri wanted to be a part of what was going on. Sorry, Siri, you have to miss out on this one. But yeah, so this all or none principle, that means there's no variation in amplitude. So we see resting membrane potential, we see it shoot up in depolarization, come back in hyperpolarization. Okay, the height of this wave doesn't change. Okay, so it's not like we can say, oh, that was a really strong action potential, or that was kind of a weaker one. It's not how it works. No variation there. But how does variation occur? It occurs in frequency. Okay, so it's not how tall is the action potential, it's how many action potentials do we get in a row. Okay, so it could be whoop, 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 kind of have this certain rhythm between the action potentials, or they can go really fast like this, right? And that would be a stronger signal. That's how the body knows the difference between a strong signal and a weak signal. Okay, so absolute refractory period. This is when we can't have another action potential because of the state of those gates. And so the cause of it, so this is during repolarization. Okay, and maybe a little bit of the hyperpolarization phase. Okay, it's caused by those two gates in the sodium channel. If you have those two gates in the sodium channel, and you get that inactivation gate that closes, when it's in that inactivated state, it's stuck there. And even if another depolarization hits it, it doesn't do anything. So it's stuck there. Okay, and that's good because there is depolarization around the membrane in that moment. Okay, so if it was able to open right up again, then you wouldn't have time to recharge. What actually would happen is we would have action potentials going both directions. Okay, because we want an action potential just to go down the membrane in a specific order. Okay, we only want it to go in one direction. We want sensory neurons to go one way, motor neurons to go the other way. If they spread both directions, that would be very wasteful for energy purposes, because action potentials are energy costly, and it would also be very confusing to the brain. It wouldn't really make sense, right? So the purpose is to create one direction of AP flow. Okay, so the way that works is if we have an axon here, and we have this part of the membrane that's depolarized, okay, that action potential is moving now to this part, okay, so that's depolarization 1, This one now starts to depolarize. This one is now repolarizing, right? But this, if we didn't have that absolute refractory period, then this depolarization would just spread back this way and cause another action potential there. But since we have the absolute refractory period, and this is in the repolarized state, and that's inactivated, then nothing happens. This doesn't cause an action potential, okay? So then this just moves on to the next part of the membrane, depolarizes there, moves on, depolarizes there. So it keeps a one direction of action potential flow. Okay, and that's kind of shown here with that one direction of flow. There's something even a little bit more specific called this saltatory transduction. What that means is when we have a myelinated axon like this, okay, with these myelination bulbs, we have these little sections in between the myelinated parts that are called the nodes of Ranvier. And those nodes of Ranvier don't have myelin, but they're packed heavily with those sodium and potassium channels, okay? And so pretty much what the myelin allows for is for that action potential just to kind of slip through the parts that are heavily myelinated and heavily insulated. Think of a very insulated wire. Okay, the action potential just kind of slips through there, doesn't need to be recharged till it hits this. That recharges it, gives it power again, recharge again, gives it power, okay? And so that's saltatory transduction. So what is myelin? It's really lipid-based, okay? It's a cell membrane of a Schwann cell just wrapped around the axon, so it's really a fat, right? And fat is a good insulator of electricity, but it's a bad conductor. So what does that do? That keeps the electricity inside of the neuron, okay? It doesn't let it spread out. The electricity tries to spread out, but it hits that bad conductor, good insulator, and it doesn't go through it, okay? Same reason we have wires in our house coated with rubber, right? Those wires that you have in your walls are coated with rubber because rubber is a bad conductor, good insulator. Okay. All right. So a couple of questions here to think about. The action potential is caused by blank of the cell membrane. Okay. So depolarization. All right. So we got either B or D here, which is primarily due to a rush of what into the cell. So depolarization, rush of what would be sodium, right? So we're D. Okay. Now a little bit connecting back to the muscle stuff that we already talked about. So what chemical goes into the neuron when an action potential arrives at the end of the neuron? So into the neuron. Didn't talk about that too much, talked about an AMP a bit, but this is calcium. Okay, so before acetylcholine goes out, calcium has to come in to stimulate that release of acetylcholine. Okay, so let's talk a little bit about that situation. Now that action potential's traveled down to the neuromuscular junction, right? The neuromuscular junction is made of three components, okay? It's made of the nerve part, right, at the end of the neuron, the space in the middle, and the muscle part, okay? So what are those called? The nerve part is the axon terminal, space in the middle is the synaptic cleft, and the motor end plate is the muscle. Okay, so those three components. What is the neurotransmitter? Acetylcholine, right? We call it ACH. Where is that neurotransmitter formed? This is always kind of an interesting part to me, in the soma of the neuron. Okay, so way up there, could be a foot to 2 feet away, right? In that same neuron, there's a little incredibly complicated network of highways that allow little motor proteins to carry these vesicles full of acetylcholine down from the soma to the motor end plate. I'm sorry, to the axon terminal. Okay. All right. So that's a little bit on that. So when this action potential arrives, right? So we're talking about that action potential, it comes down, spreads to these parts of the neuron. We're going to zoom in on one of those parts there, right? So what does the action potential in the motor end plate cause? It causes these little channels. These are voltage-gated calcium channels. Okay, just like those sodium channels were voltage-gated, calcium channels are too. So they open up, calcium goes in. So calcium in. Okay, what does that lead to? Calcium leads to acetylcholine release. Okay, so these vesicles go to the surface, release acetylcholine. Okay, what happens when that acetylcholine binds to acetylcholine receptor? What does it do? It opens up. It opens and allows for sodium and potassium to move. Just like before, sodium is going to go in, potassium is going to go out. There's a much stronger gradient for sodium to go in, so we get more sodium in, so that causes depolarization. We get an overall more positive charge on the inside. If we get enough of that depolarization, it spreads to the normal part of the membrane that contains those little sodium potassium channels we were just talking about. Notice that this is the one that has the inactivation and activation gates. These ones don't. Those are different, right? So we need to get enough depolarization to get out here and then hit those, and then we get an action potential on the muscle cell. Okay, so that action potential travels down the muscle cell. Okay, so we're going to actually start out class on Monday walking through this right here. Okay, so what I'd like you to do is try to fill it out yourself. Okay, so start in the brain. Okay, so start in the brain, go down to the steps of an action potential, go down to the neuromuscular junction events, get to what's called excitation contraction coupling, EC, right? And then get to cross bridge cycling. Okay, great. All right, a couple more key points about the nervous system and how it interacts with the muscular system. Okay, so motor units. You've talked about motor units before in other classes, defined as all of the muscle cells innervated by one motor neuron. Okay, so we have motor unit 1 coming down. You can see it branches off and just connects to 2 muscle cells. Okay, this is a bigger one. Motor unit 2 connects to 3. Okay, these are all pretty small, just so you can actually visualize them. Okay, but in actuality, we might have super fine control ones like in our fingers and our hands and in our eyes and facial muscles that might only have 2 to 5 cells. But then we have these huge ones like in our quadriceps and our vastus lateralis is a quadricep, our gluteus maximus, these big muscles that allow for large movements. That might be 1,000 or 2 muscle cells in each. And so the thing with this is it's the all or nothing principle here. So if there's an action potential that goes down that motor neuron, then all muscle cells within contract. Okay, so we have to contract all 2,000 of those if we send a signal on that neuron. We have to contract all 5 of those if we send a signal on that neuron. We can't just say, oh, let's do the first 1,000 in that motor unit, but not the second 1,000. It doesn't work that way. Okay, so that's a key point. All right, so let's talk briefly about how do we create sustained force in contractions, right? I'm having to do that as I'm standing here talking to you. You're having to do that as you're sitting. You're holding your neck up. I'm holding my back up, holding my neck up, right? How do we maintain that same force without those muscles fatiguing? Because that's a lot of force to be maintained for a long period of time, okay? And the way it happens is by rotating motor units, okay? I love the analogy here that I think of every time I'm out surfing or every time I go out to the cliffs and I see a group of pelicans fly over me. Pelicans love to fly in this V pattern, right? But if you watch them closely and for long enough, which sometimes is hard to do because they're going fast and they go past you, what you'll notice is this guy in the front, or girl, eventually will make his way back. Okay, he'll move back one, and then this guy comes up and leads the charge. Okay, eventually that guy gets tired, he comes back, this one moves back again, and then this one comes up and leads the charge. Okay, and this constant rotation happens, right? Why? Because it's way harder in the front because of wind resistance, right? This is, you know this if you're a cyclist. If you watch the Tour de France at all, you know this is why teams in the Tour de France are so important, because that person who wins is only out at the very end, only out in front at the very end. This team is carrying them along the rest of the time. And this is exactly how the motor units work. Okay, so we might have one group of motor units holding up my back muscles for a little bit. They start getting tired. They start sending some chemical signals that the brain picks up on through sensory neurons. And the brain says, all right, those ones need a break. Let's let those relax, and kicks in another group of motor neurons, right? Motor units. And it just rotates like that. Now that other group is resting, recovering, and then the other one can come back. So that's kind of a cool function of how that works. Okay. Last point here is, how does the brain vary how much force is created by a muscle. Okay, and there's two levels of this. Okay, so first, there's the signal strength, really, which means frequency, right? From the higher brain centers to the primary motor cortex. That's going to dictate how many motor units contract. Right, so we had that signal up in our front prefrontal cortex that says, all right, I need to contract. I need to contract my shoulder muscles to reach out to my keyboard, right? Okay, so that might be a small signal. So low frequency coming from the prefrontal cortex, and that goes to a small amount of motor neurons in the primary motor cortex. So that activates a small amount of motor units. Okay, but I could use that same deltoid muscle that I'm going to use to reach my shoulder out. I could use that same deltoid muscle to throw a punch, right? When I'm using my punching bag to do a workout, I can punch it as hard as I can. I'm going to have to use a lot more motor units for that, right? So the signal from the higher brain is stronger, higher frequency, that spreads to more motor neurons in the primary motor cortex. So we activate more motor units and we contract more muscle cells, okay? So that's number one. And that signal strength also kind of dictates which motor units contract, right? Which ones. And it always works this way. It's always the small ones first, and then gradually larger and larger stimuli, or larger and larger as the stimuli increases. So we don't not contract the small ones. We contract the small ones first, and then small ones and big ones, and small ones and bigger ones, like that. Okay, so that's level number one. Okay, level number two is within one motor unit, that frequency of action potential can change as well. Okay, so down that one motor neuron, we can send a low frequency action potential, which would result in a low amount of acetylcholine release, which would result in a low frequency of action potentials on the muscle. Okay, but if we want to contract that muscle really hard, that signal from higher up is going to be stronger. It's going to lead to a stronger action potential on the motor neuron, and that's going to lead to a lot of acetylcholine release, which leads to a lot of calcium release. And how does that make us stronger? Well, it opens up more myosin heads, right? If we've got more calcium released, we got more calcium binding to troponin, more open myosin binding sites, and that's how we're getting our force from muscles. Every new myosin head that contracts gives us more force, right? So just briefly, let's look at this here to kind of show you what I mean. Okay, so we've got some neurons in the prefrontal cortex. Okay, we've got our muscle, and we've got our primary motor cortex. All right, and then we've got our muscle. So we have a group of interneurons, right? Those are neurons going from neuron to neuron between the higher brain and the primary motor cortex, and we're skipping that third neuron here just to make it simple. Okay, and so we've got that group, and we have a group of motor neurons in the biceps brachii, for the biceps brachii, right? And those are all pooled in the same part of the primary motor cortex. Okay, so we have a low strength stimulus, right? We're just reaching out to our keyboard with that anterior deltoid. So what happens? We activate a few neurons in the prefrontal cortex. And on those few neurons, we have pretty low frequency of action potentials. That's going to activate a small bit of the pool of motor neurons in the biceps brachii. It's going to activate those. So smaller units stimulated, low action potential frequency on each, right? So it's going to lead to decreased calcium release. It's going to have some, not decrease, but low calcium release. Okay. All right. So that's with the low frequency stimulus. So what happens the other way? So we have a high frequency stimulus. I go to punch that punching bag, right? You go to punch it. What happens? We get more higher level neurons that get activated. On each of those neurons, we have a higher frequency stimulus that activates more motor neurons here. So a larger part of that pool of motor neurons in the muscle. And on those motor neurons, we have a high frequency of action potential on each one. So it leads to increased calcium release and more force. Okay, so good thing for you to be able to walk through and understand. And I'm going to leave this part to you. Okay, so you're going to go through and just fill this in. And the example here will be using your pectoralis muscles to type. So you're kind of reaching across your keyboard typing versus using that pectoralis muscle to throw a big punch into the punching bag. Okay, all right, so work through that. Let me know if you have questions on that next time we meet. All right, guys, have a great weekend. We'll see you soon.